Hi, and welcome back. This is Botany 101, and my name is Angela Nishimoto. I am a teacher for this class. Hello. Okay. Today we're going to be doing molecular genetics, okay, which has to do with the synthesis of proteins. Okay, our objectives for today. First of all, we're going to talk about what the genetic material is, okay, in molecular genetics. Because remember last time we talked about genetics. We're also going to talk about some of the discoverers and some of the pioneers in molecular research, such as Watson and Crick and other people. Thirdly, we're going to talk about the genetic code, all right? So the genetic code, of course, is vital, okay, to the process of making proteins, all right, and the expression of genetic qualities. We're going to talk about transcription. We're going to talk about translation. And lastly, we're going to talk about a phenomenon known as mutations. Okay, for one thing, okay, the genetic material is called DNA. If you remember, DNA, okay, our DNA as well as plants' DNA is located in the nucleus of our cells, okay, plants and animals both. And generally DNA, okay, is sort of like a molecule that has one strand, and it has a second strand, and it's sort of like a rope ladder that's been twisted with hydrogen bonding occurring between adjacent strands, right? So at each place you have these nucleotides, right, that actually make up the DNA molecule, and you have the nitrogenous bases that do hydrogen bonding. Remember hydrogen bonding that we talked about quite a long time ago, yeah, at the beginning of the semester. Now this is like a space-filling diagram showing, you know, a little piece of the DNA molecule, which is pretty, actually is pretty big, okay? So you can see that you have carbons and hydrogens and oxygens and etc. Okay, just the molecules of life. So remember, DNA is a kind of nucleic acid, one of the 4 types of molecules of life. Remember, carbohydrates, lipids, proteins, and nucleic acids. Okay? So what are genes? Because of course we're talking about genes. Remember last time we also talked about, you know, classical genetics, which the unit of inheritance is called a gene, okay, or etc. So genes actually code for traits, and the genes are located on chromosomes. Remember chromosomes? Okay, chromosomes are made up of DNA plus proteins. Okay, so these actually come together, and before they didn't really know what proteins were for, but now they're thinking that maybe proteins that make up chromosomes might have a regulatory function, you know, turning one gene off, turning another gene on. Yep, all right. So in the past, they actually asked, since chromosomes, you know, are actually passed on physically from one generation to the next, in the past, they didn't know that DNA was the molecule of genetics. They didn't know whether it was DNA and protein. So which would they have thought? They probably thought, you know, a lot of people did believe, the most brilliant scientists thought that proteins, because proteins, you know, are very complicated, remember? Very complex and very large molecules, and people thought that nucleic acids were much too simple to really account for, you know, the myriad of genetic expression, you know, in terms of phenotype. Remember phenotype, what an organism looks like, right, which is the expression of the genotype, okay? Okay, so finally, this controversy about which was genetic material. So chromosomes, mitosis, meiosis wasn't actually delineated until the end of the 19th century or the beginning of the 20th century. It wasn't until Hershey and Chase did their very elegant experiments in the 1950s that people knew for sure, okay, what it was. For one thing, they used very simple organism, okay, they used bacteria, okay, called E. coli, Escherichia coli, which is a coliform bacteria. And this bacteria, you know, has a tendency to be infected with certain viruses, which are known as bacteriophages or bacteriophages. Phage means to eat, and these viruses actually feed on bacteria, okay? So these viruses, okay, viruses, remember, I don't know if we talked about this before, but viruses are made up of a nucleic acid with a protein coat. That's basically what a virus is. So viruses are not cellular. Therefore, viruses are not considered to be alive, because remember when we talked about living things such as plants, such as fungi, such as animals, generally all these different kind of organisms, even bacteria, have cell components. They have a cell membrane which borders a cell, and they have a cytoplasm, and they have genetic material, right? So anyway, so these bacteria can be infected by these viruses, which are called bacteriophages. Okay. Now, okay, so we can kind of look on the PowerPoint, and what we have here, these are illustrations of bacteriophages, okay, bacteriophages. So you see that generally the genetic material can be either DNA or RNA. A very famous kind of virus that does have RNA as its genetic material is a retrovirus that is responsible for AIDS, apparently, according to how people delineate that now. But generally, the HIV is human immunodeficiency virus, okay? So it's actually an RNA virus. So you can have either DNA or RNA as a genetic material, then you have protein, okay, that makes up the rest of it. So viruses are not cellular, despite the fact that they can give us things like the common cold or influenza or, let's see, hepatitis or any one of a number of pretty bad diseases. Their viruses are very simple. But generally, viruses are not alive. They have no metabolism. They don't actually have any kind of biochemical reactions happening with them unless they have infected a cell. They need to actually infect a cell to allow them to multiply. Okay? So Hershey and Chase use bacteria and they use viruses for their experiments. Okay, so what happened is that, so generally viruses, to reiterate, are nucleic acids with a protein code. Okay, so what Hershey and Chase use, they use radioactive isotopes. Okay, remember isotopes? Isotopes are variants on like a normal kind of atom. So they use radioactive sulfur. Okay, they use a radioactive sulfur to label or make radioactive the protein code, and they use radioactive phosphorus, okay, in order to label the nucleic acid. Okay, because remember, right? Nucleic acids are made up of nucleotides, and nucleotides, what are their components? You have a sugar, you have a phosphate group, which has phosphorus, and you have nitrogenous bases. So those are the three parts, okay, main parts of a nucleotide. Nucleotides are linked together, and what they do is they make the nucleic acid, okay, the molecule. Okay, so generally, only the virus's DNA actually enters the bacterium. Okay, bacterium is singular for bacteria, which is plural, all right? So this is a very nice illustration showing pretty much the basics, and Hershey and Chase did what they call a very elegant experiment. That means it's very simple, uses relatively simple, components, and they can actually show something or support a particular hypothesis in a very strong manner. So this is actually the experiment that concluded that people could definitely conclude that DNA or RNA is genetic material, which are nucleic acids. So what did they do? They had a bunch of Escherichia coli, E. coli, which is a bacteria that comes from our gut. Okay, it's a part of our normal gut flora. And the viruses they used was a phage virus, a T2 phage. Okay, so what does it do? It actually infects the virus. So what they did is they actually labeled, in one experiment, they labeled the DNA, which has phosphorus, with radioactive phosphorus, because all organisms or even viruses will, as they're replicating, they'll take up whatever raw materials, whatever elements are in their environment. It treats radioactive components as well as regular phosphorus, radioactive phosphorus are actually taken up in the same way. So generally, they actually let these viruses, grow in media, actually reproduce. And what happened is that, this one here had its DNA labeled. So what they did is they let them sit for a while until they had generations of E. coli that had been infected with the DNA from the virus. And then what they did is they just put it, they put it in a regular kitchen blender to knock the phages off the surface of the bacterium. Okay, then they centrifuge it, because what you do when you centrifuge something is that the particles inside the medium or the liquid will actually settle out. The densest material settles to the bottom, the lighter or the supernatant kind of floats on the top, and the viruses, despite these illustrations, are very, very, very much smaller than the bacterium. Okay, the virus is probably about 100th to 1,000th the size of a bacterium. So what they did in this case is they centrifuge it, and they have a solid pellet with the larger component at the bottom, the supernatant. Okay, so what they did is they used a Geiger counter to find out that it was a pellet that had the radiation. Okay, that means that the nucleic acid was actually passed into the bacterium. A second, reciprocal or inverse kind of scientific experiment was done with them, where they used radioactive sulfur, and that labeled the protein code, because a lot of amino acids, remember, amino acids are the components making up a polypeptide, which makes up a protein. So what, excuse me. So what they did is they actually took the same thing, except they had a radioactively labeled protein. They did exactly the same thing. Put it in a kitchen blender to remove the phage, from the phages from the surface of the bacterium. They spun it in a centrifuge. Again, what they found out is that the supernatant, or the liquid with the smaller particles, actually were radioactive. So what they were able to show then, that it was actually the nucleic acid. In this case, that was a molecule of heredity, because that was actually passed on from one generation of the E. coli to the next, All right, so that was what they call a very elegant, very simple experiment. Now, so after they did do that, the race to find out the structure of DNA was on, because at the time that people were working in the 1950s, this was about the time that electron microscopes were just coming into being used in the really big ticket kind of laboratories. So Watson and Crick did some work in the 1950s. So this is a photograph of James Watson. All these people that we're showing you, and all the people who did these different kinds of work in biology, they're all brilliant people. James Watson, very brilliant man. At one point, he was the head of the Human Genome Project. This is Francis Crick. Francis Crick unfortunately passed away a couple of years ago. Another very, very brilliant fellow. Kind of eccentric, but what a brain he had, and he actually went on to do work on artificial intelligence. Now, Watson and Crick, part of the data that they used, so they're very famous. They actually were the people who outraced Linus Pauling and other big labs all around the world to actually show the public, and the scientific public also, the structure of DNA. And this is actually the type of some evidences that they use. This is actually what you can see when you do something called X-ray crystallography. It is a very painstaking sort of technique that people could use. So there was another PhD person, professor, not a professor, but a researcher named Rosalind Franklin. She worked in Maurice Wilkins' lab over at Cambridge. I believe it was Cambridge. Because James Watson is American, but he went over to England to do this work, to be in these labs and do this basic research. Now, generally, he was very young too. He was only like about 22, 23, something like that, when he and Crick got together. And I think Crick was probably in his mid-30s at the time that they partnered up. So what Rosalind Franklin did, and what people who did X-ray crystallography would do, they'd actually extract or get the material, the chemical that they were interested in. They'd crystallize it, very, very painstaking, very, very slow technique. It could sometimes take a week or maybe even more to actually make a good-sized crystal that you could do. And then what she did is she bombarded it with X-rays, and that showed a refraction pattern. And this refraction pattern is something that she had, as her work. So she was kind of working on that. One day, because scientists are usually, actually, you think of somebody in an ivory tower doing, mad scientist experiments all by themselves. That isn't the case. Successful scientists, they talk a lot to each other. They're very sociable people. They can actually work on their own, but they actually also have to do teamwork too. So he just happened to be over at Watson and Crick. We're visiting over at Maurice Wilkins' lab, where Rosalind Franklin worked, and even the double helix, which is a book written by James Watson. He talks about this. He kind of sidled over. Rosalind Franklin was not at her desk, so he kind of peeked and saw what was there. When he actually looked, he looked at the X-ray crystallograph of DNA, and then he and Crick realized that the molecule was symmetrical, that it was the same width all the way up and down. And what he actually did was he actually looked at an X-ray crystallograph, and and that is something that you really have to really know structure and everything like that to infer the structure of the molecule, because at that time they weren't actually able to take photographs or even to look at a molecule. But they didn't really have the technology at the time. Using that evidence, as well as something called Chargraff's rule. Chargraff was a chemist. Chargaff, and what he did is he actually took apart DNA, and he figured out that the number of adenines equaled the number of thymines. He also figured out that cytosines equaled the number of guanines. Okay, so that was very, very important information. So Watson and Crick's, brilliance, their brilliance was actually taking small pieces of the puzzle, putting them together, okay, to make a very large generalization, which is very impressive. Just astounding. These men were brilliant, are brilliant. Watson is still brilliant. Anyway, so Watson and Crick. Okay, so from what they did, they actually were spending their time building models, and people laughed at them. They thought, this is what introductory chemistry and organic chemistry students do. They actually get the ball and stick, and they actually build models. And Watson and Crick were actually doing that in their lab. They were laughed at. Okay, so they knew from Rosalind Franklin's crystallograph that the DNA was actually a molecule that was symmetrical all the way up and down, and they also knew from Chargaff's root too. They actually thought, okay, that, we'll talk about this in a little bit, that remember that DNA will form a double helix, and a nucleotide is made up of a sugar, in this case deoxyribose sugar, a phosphate group, and a nitrogenous base, okay, each nucleotide, which is a component of the DNA. Okay, it's a unit of a nucleic acid. Okay, so the bases, okay, you have adenine, or A, thymine, or T, guanine, or G, and you have cytosine, or C. Okay, so the A and the G, the adenine and the guanine, are sort of nucleotides which they refer to as purines, okay, purines. And they generally have two ring structures making them up, okay, the bases that is. And thymine and cytosine are other kind of bases with a single a ring structure, and they're called pyrimidines, okay. So remember that Watson and Crick, they had never seen the molecule. They can also only use inference, okay, from data that they had. Okay, so A always pairs with T, or a purine, a double ring structure always pairs with a pyrimidine. So A is a purine, T is a pyrimidine. G, or guanine, is a purine, cytosine is a pyrimidine, because they knew that the molecule was symmetrical. Okay, they believe, well, first of all, they thought that maybe the sugar phosphate backbone, okay, sugar phosphate, was on the outside of the molecule. But if that is, you'd have bulges, and like that. So what they figured, put it on the inside. So at first they thought the purines would pair together and the the pyrimidines would pair together. But they knew that if that would happen, you'd have these big, also bulges sticking out. Okay, so they were correct in terms of the inference. So here is showing that you have this adenine, this double ring structure, pairing with a thymine, a single ring structure. Not just that, these red lines here, they're actually showing the two hydrogen bonds that hold these two bases together. And then between the guanine and the cytosine, you have 3 hydrogen bonds. So they, in all their brilliance, they were able to infer the structure of DNA, okay, just from, because they didn't have diagrams for sure, they didn't have a photograph of the molecule, okay, so they were able to get inferences, or taking evidence from different pieces, forming, a big overall generalization, which was brilliant. Okay, now DNA, okay, does replicate. Remember, this happens during the S phase of the cell cycle. Okay, and what you have is complementary base pairing. So one of the things, very famous sort of understatement that we're going to talk about in a little bit. So here is you're actually showing correct pairing between adenine and thymine, and guanine and cytosine. Sometimes though, if the cytosine is in a different kind of form, you can actually have a mispairing. You actually have a cytosine pairing with an adenine, or you can have a thymine pairing with a guanine. So this is something that is like a mistake in base pairing, and usually when you have this kind of mistake, it's called mutation. Okay, we'll get back to that a little bit later. But here we show the structures of the two purines, adenine and guanine, and we have cytosine and thymine. Now this uracil is a base that is in RNA. We'll talk about that in just a couple of minutes. Okay? So generally, this complementary base pairing, if A always pairs with T and C always pairs with G, okay, that actually is something that Watson and Crick in their paper said that we have not overlooked the fact that the structure, the proposed structure of deoxyribose nucleic acid suggests a copying mechanism, because this is something that people didn't know. They're thinking, how was it actually transmitted from one generation to the next? They didn't really know that, even in the 1950s. Okay, so they were able to brilliantly figure this out. Okay, so what happens when DNA unwinds? Okay, for DNA will replicate. First of all, the DNA unwinds because remember it's in a helix, which is a spiral. The DNA unwinds, and each strand, you know, separates from each other, and each of them acts as a template, okay, for a new strand. All right. So you have this thing coming. So this is what they call semi-conservative, because in every new strand of DNA, you have one old strand and one new strand. So it is semi-conserved. Semi means half. Conservative means that you hold on to one part. Okay? So this is showing DNA. Remember that this happens during the S phase, right? Before mitosis, before meiosis, right? So the strand unwinds, and what happens is that you have these free floating bases, you know, nucleotides floating around in the nucleus, and they'll actually come in and they'll do covalent bonding between adjacent phosphate groups and sugars, which form the backbone, okay, the outside of the molecule, and then the bases will pair, okay, they will hydrogen bond. Okay, it seems like it's relatively simple, and it really is, and it's a miracle, you know, that our bodies can do this. Plant bodies, of course, do this too. All right. So what you'll see is that generally, when you have DNA replication, you always have one old strand in one molecule, one old strand in the other molecule, and you'll have one new strand. Okay, in each new molecule, and this happens, you know, when the chromosome replicates itself. So then you'll have two sister chromatids. Remember that we talked about that a couple of times before, yeah? Okay, so think about this. Okay, so when you're tested on this, you will probably have to answer a question like this: what would be the complementary DNA sequence of bases, okay, to this particular sequence of nucleotides? Okay, you have TCG, TAC, CAG. Okay, so what would pair with T? A, right? What would pair with C? G. What would pair with G? C, and etc. So you'd have A, G, C, A, T, G, G, T, C, right? This new strand being complementary to the other. If T pairs with A, C pairs with G. So that happens almost all the time. Usually in terms of bacterial genetics, okay, there's only maybe 1 in a million or 1 in a billion mistakes. Okay, only 1 in a million to 1 in a billion mistakes. So that is actually really, really good. So our cells, you know, and the cells of plants do an excellent job, okay, in terms of making sure that all the information is as it should be. Okay. Now the next thing is that generally genes control proteins. Okay, so what is a protein? Remember, so protein is one or more chains of the subunits known as amino acids. Do you remember amino acids, right? Amino acids are made up of, right? They're actually made up of, right, central carbon, remember that? And we also have the fact that you also have this hydroxyl group, right? COOH, you have an amino group, or NH3 or NH2 on the other side, you have a hydrogen, and then lastly you have an R group, okay? Anyway, so you have this thing that you have proteins, okay, made up of amino acids, okay? And when you have a string of amino acids that are they're usually about 50 to 500 amino acids long, you have something called a polypeptide. Remember, poly means many, right? Peptides refer to the amino acids or the subunits of a protein. Okay, so remember too the 4 levels of protein structure. The first level is just a chain of amino acids. The second level is when you have nearby R groups. Remember the R groups, right? And what they're doing is they're hydrogen bonding. Okay. Anyway, so let's see. So you have polypeptides. Okay. And then what happens is that you have the fact that different kinds of proteins have different kind of functions. All of these are very, very important. Okay. You have enzymes. Enzymes help to facilitate chemical reactions. Biochemical reactions generally do not happen without the presence of enzymes, and there are thousands of different kinds of enzymes. Okay? We also have structural proteins, such as in our bodies, we have our skin has keratin, our nails, our hair has a protein called keratin. We also have collagen that helps to keep your face from sagging, for another thing. Okay, you have elastin that allows you to make, you know, faces and to actually stretch your skin and things like that and bounce back, you know. Anyway, so we do have structural proteins. All right. You also have regulatory proteins because some hormones are proteins and they serve a regulatory function. And then lastly, we also have transport proteins. Remember when we have the cell membrane, you have the phospholipids all kind of oriented this way. You have a whole bunch of them, right, making up the membrane with proteins embedded in there. And proteins also serve as a way for different sort of materials to go from inside of the cell to outside of the cell, or from inside an organelle to outside an organelle. Okay, so you have all these different kinds of proteins. They're very extremely important to life. Okay, we couldn't live, okay, without proteins. Right, and this is showing you here, okay, on the PowerPoint, that just a reminder, these are different depictions of a particular, I think it's triisomerase, one of those enzymes, to show you that proteins are indeed very large molecules and they're very, very complex. Okay, so we can, imagine why it was that early workers believed that proteins were the molecules of inheritance, just because it seems that inheritance is much too complex a sort of thing to happen for just something as simple as DNA or RNA to actually facilitate. So we're going to find out right soon, okay, how they actually have a role to play, okay, in terms of the manifestations of our bodies, what we look like, and etc. Okay? All right, so we find out that genes that are written on the DNA in a way, okay? And so genes actually control the manufacture of proteins. So remember that proteins have definitely an extremely important function in all the different kind of biochemical reactions that happen in plants' bodies as well as in our bodies too. Now two scientists, another couple of brilliant fellows, okay, George Beadle and also Edward Tatum. Okay, very important fellows here. Okay, they look very serious. Many scientists are pretty serious, yeah, but still, a lot of them are very congenial with each other and like that. Yeah, so I think that I remember that Watson in his book, The Double Helix, talks, about them going to the pubs and kind of talking things over and like that, because he lived in England for a while. Cambridge, exactly. Okay, so what they did is they actually did work on a fungus. Okay, I believe it's an ascomycete called Neurospora crassa. All right. So what they did is they actually got this fungus and they bombarded the fungus, various funguses with fungi with X-rays. We know that X-rays can cause mutations. So what they did is they actually got these mutants from of this fungus, and this is actually one that's reproductive. I believe that these are their spores. Okay, so this is actually sort of like a kind of a very beautiful, fancy picture of them. Okay, so this is actually a picture of the organism, Neurospora crassa. So what they did is they bombarded, these fungi with X-rays, and then what they got is mutants. And these mutants would have mutations that would interfere with their metabolism. From this, they formulated the fact that they had the one enzyme, one protein or one enzyme, one gene sort of idea. So what they did is they were able to kind of figure out which enzymes, were actually coded for by the DNA, in this particular fungus. Okay, that's kind of like a famous thing. So into the 40s, into the 50s, and into the 60s, people, did a lot of work with nucleic acids and with proteins. Okay, so this is like another little puzzle piece that helped people to figure out how exactly proteins come from nucleic acids, okay, or how nucleic acids code for proteins. Okay, so what they do is that the bases of DNA, the A and T and C and G, the sequence of bases will actually affect the sequence of amino acids, and we're going to get into that soon. But first, okay, so we have DNA. What does DNA do? DNA in the nucleus is like the master blueprint. Okay, it resides in the nucleus. DNA never leaves the nucleus, okay, as long as the cell is alive, all right? So when you do, like we're going to talk about next time, we're going to talk about molecular DNA technology. If you want to sequence DNA, one of the things you have to do is to rupture and kill the cell so that you can get the DNA out of the nucleus, okay? Anyway, so deoxyribonucleic acid will code for a special kind of nucleic acid called RNA, ribonucleic acid, okay? Especially in the form of mRNA or messenger RNA. Okay? Then the messenger RNA will actually code for the different kind of amino acids that make up a polypeptide, okay, which will eventually make up a protein. So generally a good thing to remember that the flow of genetic information from DNA, the information goes to RNA, another kind of molecule, and that will actually code for amino acids that make up a protein. Okay, so DNA codes for RNA, codes for protein. So RNA, of course, is ribonucleic acid. Okay, so there are differences between DNA and RNA. Okay, so there are 4 differences that I can kind of think of. For one thing, RNA itself has a different sugar. Okay, deoxyribonucleic acid has a deoxyribose sugar. RNA has a ribose sugar. There's a little bit of a difference. So, RNA has a different sugar. You don't really have to know specifically what the difference is. It's just that DNA, actually the sugar actually is missing an OH. It actually doesn't have the OH, whereas a ribonucleic acid sugar does. RNA, furthermore, is single-stranded generally, okay, in eukaryotes. All right, whereas DNA is double-stranded, okay? Thirdly, RNA tends to be a smaller molecule, okay, because generally the DNA that codes for the RNA only codes a little bit, okay, of the genetic code on the DNA, okay, will code for the RNA. And another thing is that RNA uses uracil, okay, as a match for adenine instead of thymine. Don't know why that is. I've actually asked the chemist, I think an organic chemist, about it. He said he doesn't know. Other people have said, well, that's just what happens, you know. They don't know why necessarily. One of the things to remember is that scientists don't usually ask why, because that usually questions for intention, you know. And scientists, you know, scientists deal in empirical evidence, just with things that you can sense. Now, for an RNA, then, A generally will pair with U, and like in DNA, G will pair with C. Okay. Okay, here's a diagram showing, so here's a double helix of a DNA molecule, deoxyribonucleic acid, okay, and the bases, of course, are cytosine pairing with guanine, adenine pairing with thymine. Now the difference is, we can see 2 differences here because we don't really see the sugar and etc. But here you see that RNA tends to be single-stranded, okay? Remember that RNA actually forms off a DNA template, okay? So we'll get into that in just a sec. So you have the 4 bases of RNA, cytosine pairing with guanine, and adenine pairing with uracil. You can see, look at uracil and look at thymine. They're very similar in structure, yeah? Okay, so uracil in this case will pair with adenine, and guanine will pair with cytosine, okay? Now, okay, so the genetic code, okay, does have to do with RNA as well as with DNA. Okay, so we're going to get into the different types of RNA, all right? So first of all, we have mRNA, also known as messenger RNA. So what does messenger RNA do? It actually carries a message. It carries a message that's coded for in DNA, making a complementary copy of RNA or messenger RNA. It will carry it, you know, to where it actually does the protein synthesis, okay? So this is showing, you know, a cell, and there's of course a cell membrane, and here you have the nucleus of that cell. Remember, the nucleus has a double membrane around it with nuclear pores in it, so things can move out or things can come in. But only certain things can leave the nucleus, and DNA is not one of them. Okay, DNA stays in the nucleus. So this is the DNA. Okay, it actually unwinds. All right, and then what that produces is single-stranded RNA. You'll actually have messenger RNA made by the process known as transcription. They call it transcription because you're still in the language of nucleic acids, right? You have DNA and you have RNA. These are 2 different kinds of nucleic acids. So when you actually see one of those old movies, Perry Mason or Dragnet or something like that, where you see people in court, Dragnet was more like a police show. But anyway, so if you see one of those old movies or old television shows where people are in court, you have, you know, one person giving their testimony, right? And you have somebody actually over there typing really, really fast, right? That person is a transcriptionist, but they're still, you know, speaking in English, as far as we know, in an English American court, right? So you have the same language, DNA in the language of nucleic acids coding for RNA, which is still in the language of nucleic acids. Okay, so here you have a single-stranded RNA being made from a DNA template. Okay. Next thing that happens generally, okay, we're going to go through this again, but the next thing that happens is that the messenger RNA usually has parts in them called introns. Introns are non-coding regions, okay, of RNA. Usually what happens in the nucleus is that the introns are spliced out, and then the rest of the gene is, the RNA is actually joined back together. What then happens is that the mature messenger RNA leaves the nucleus and goes into the cytoplasm. And what happens then is that in the cytoplasm, okay, this is kind of complex, but you have this messenger RNA, okay, joining with something called, remember, a ribosome? The ribosome is a small little, tiny little, speckles that are inside of the cell. Remember when we looked at the ER, the endoplasmic reticulum? You had all those little dots on it. Okay, so each of those little dots is a ribosome, right? So the ribosome and the messenger RNA will form a complex. Then you'll have another kind of RNA known as transfer RNA. We're going to talk about that next, okay? And then anyway, so at the ribosome is where the mRNA, okay, the message in the mRNA is translated into the messenger protein. So we'll get into that in just a sec. See, on this side you have like the growing amino acid chain on this side of the transfer RNA, okay? All right, so transfer RNA, okay, is also RNA, of course, single-stranded. Okay, it actually has 2 sites on it. So generally the transfer RNA, you can see it's pretty complicated. It's not that big of a molecule though. But here you have, the fact that you have a single strand, and you have the single strand of RNA bent and folded and hydrogen bonding into a particular kind of conformation. Okay, on one side of the transfer RNA, you have a site, a binding site for an amino acid. Each kind of transfer RNA will actually only have one type of amino acid on one side. On the other side of the transfer RNA, you have something called an anticodon. Okay, it's actually going to be complementary to the codon. Okay, and the codon is on the messenger RNA. Okay, so we're going to get that into that in a little bit. The third type of RNA is called ribosomal RNA, or rRNA. It is a component of the ribosome, okay, along with proteins. And remember, the nucleolus, right, in the nucleus is responsible for the manufacture of ribosomal RNA and the ribosome itself, or the other components, of the proteins of it. So this is a diagram of the structure of a ribosome. Generally, the ribosome has 2 subunits. This is funny, but the small one is called a small subunit, also known as SSU, and this one here is called the large subunit, or LSU, okay, large subunit. If you have to take genetics, you'll probably abbreviate it like that, okay? So you can see different viewpoints of it. So you think all those little dots that were inside of the cell, each of them is pretty complicated, but still. Okay, so you have the small subunit here, which is colored in blue, and the large subunit, which is colored in red, okay, in this particular illustration. And they'll have binding sites. Okay, they'll have binding sites, okay, for the messenger RNA, and a place where you can have a place for the tRNA to be too, on the ribosome. So even though they're really, really tiny, they're still pretty complicated. So, life, living things, we actually are a lot more complex, different kinds of animals and plants, much more complex than we might think. Another thing is, the thing is when you're doing a dissection, like cutting apart part of a plant or part of an animal, looking at subcellular structures and things like that, somehow, even though it's fascinating to look at the components are never as great as a whole, as something that's living. Just like when you catch a fish. Fish are really beautiful when you see them in the water. You catch a fish and it's dead. It just never looks as beautiful as when it's alive, That's something really true, okay? All right, then, so we have the genetic code, okay? So the genetic code is read in units called codons, okay? Codons are merely 3 consecutive bases. All right, so it could be ACG, ATG, depending if that's in DNA. Oftentimes they'll have a genetic code, okay, actually transcribed into the RNA codons. If you open your textbook into the genetics chapter and you look at table 7.2, okay, you'll actually see the genetic code written out. So the thing is miraculous, okay? So each of the codons, and there are 64, right? There are 64 different codons, will generally specify a specific amino acid, okay? So there are 64 codons, and 4 of them actually code for other things. So 61 code only for amino acids. So you're thinking, wait a minute, aren't there only 20 amino acids? That's true. Okay, but we'll get into that in just a second, just talk about that. But 61 of these codons code for amino acids only. One of them, AUG, and this is in RNA language, you can see because of that U in there, the uracil. AUG is actually the start codon. It will actually tell these components, making the protein, that now it's time to initiate protein synthesis. Okay, another thing too is this start codon, AUG, also codes for methionine, MET. Okay, so methionine is one of the amino acids, okay? And the start codon, and that is actually the only codon that codes for methionine, even though it also codes for a start codon. So all proteins generally in plants and in animals start with AUG to kind of tell the different components of the cell, this is now time to start making the protein. Now there are 3 what they call stop codons. That means that when, after you initiate and then the polypeptide elongates, and then these 3, UAA, UAG, UGA, these are the stop codons. Okay, they'll actually tell the components to stop making the protein. Okay, so now for all the 64 different kind of codons, don't be all worried and think that I'm going to make you memorize all of them, but you do have to know AUG is a start codon, and that UAA, UAG, and UGA are the stop codons. Okay, those are the only ones you need to know. Just know also that the 61 will actually code for amino acids. Okay, all right. Okay, then. Okay, so furthermore with the genetic code. Okay, so remember the red codons, a sequence of 3 consecutive bases adjacent to each other. All right. Okay, so when we look at an illustration like this is showing a particular section, okay, of RNA, and you can see in here, in this case, we have a codon 1 with its GCU. And if you were to look onto your little table, okay, in your textbook, if you look up GCU, that codes for a particular amino acid known as alanine. Okay, now ACG will code for threonine. Okay, GAC, okay, GAC, okay, will code for aspartate or asparagine, depending on what form that particular structure is in. So each of these codons will actually code for a specific amino acid, each sequence of 3 bases. Okay, this is one of the things that we know, that if you actually have some kind of mistake, if you have a mistake in copying, sometimes what can happen, when DNA codes for DNA, you have DNA replication, what can happen is that's called a mutation that can actually call for the wrong, so-called wrong amino acid to be put in there, depending on the mutation. Okay? Alrighty, so the genetic code is redundant. Remember that there are actually 61 different codons that will code for, okay, that will actually code for certain amino acids, but they're not ambiguous. That means sometimes you'll have 4 or even more different kinds of combinations making up a codon that actually code for only one type of amino acid. But it is not ambiguous. That means if you have, say, CGU, it will always code for arginine, always. Okay, and just like GGU, that will always code for glycine. Okay. Another very important point, okay, so the 2 important points here. The genetic code is never ambiguous. Another thing that's important in terms of application or technical use of this is that the genetic code is universal, okay? This is one of the reasons why you can actually take genes, okay, from a jellyfish, put it into a tobacco plant, okay, or genes from a jellyfish, putting it into a mouse, and have the mouse glow in the dark. Okay, a phosphorescent mouse. And they had these over at the Bishop Museum. They had a few years ago, they had those recombinant mice, made in that lab over at UHM. And another thing that, one of the icons, okay, of DNA technology, they have a tobacco plant that they put firefly genes in, which also phosphorescence. Okay. All right, so that's one of the things, is that genetic code is universal. You can actually have gene from a human being, put it into a bacterium, okay, and then the bacterium will actually produce a protein, a human protein. And this is something we're going to see later on too, but it's actually responsible for the production of synthetic insulin for people who have, type 1 diabetes, who need to inject themselves with insulin every day. Okay, so technical application, all right? So because the genetic code is universal, the genes in a bacterium, in protists, in fungi, plants, and animals, okay, they can all be put into different kinds of organisms. You can put genes from a bacterium into a fungus. You can do vice versa, take a gene from a fungus, put it into a plant. Okay. You can take a gene for pesticide resistance, also insert it into a plant. You can you can actually make a plant make its own insecticide. Okay, that is one of the controversies in terms of genetic engineering. Okay, so we'll get into that next time. But this is a very important thing. This is why there's been an explosion, okay, in molecular biology and also the different products, okay, of genetic engineering. Okay, to do an overview, okay, of the two parts of protein synthesis. So here's another pretty good illustration. Okay, first of all, you have the double-stranded DNA coding for RNA. Okay, and the RNA could be messenger RNA, it could be tRNA, or it could be called rRNA. Okay, right? Messenger RNA, transfer RNA, or ribosomal RNA. But generally, okay, this transcription, strictly speaking, is generally for messenger RNA. After the messenger RNA is processed or the introns are spliced out, you have what they call the exons, or the coding region, okay, of the messenger RNA spliced back together. The messenger RNA will leave the nucleus. There it will actually latch on to a ribosome, the small subunit of the ribosome. Okay, then you'll have the large subunit coming in along with a transfer RNA, okay, which we don't see here. Okay, but generally what happens then, you have the transfer RNA being the site where, okay, the transfer RNA will allow bring in the amino acid, and the anticodon part of the transfer RNA, remember the transfer RNA has the anticodon, that will actually bond, okay, bond to the codon that's on the messenger RNA. Okay, then what will happen is that a second transfer RNA will come in, bringing in the second amino acid. The second transfer RNA will actually have its anticodon bonding to the codon, okay, of the messenger RNA. Then, this is important, between the first amino acid on the first transfer RNA and the second amino acid on the second transfer RNA, you'll have a peptide bond being formed. Then, on the first transfer RNA, okay, the bond between the transfer RNA and the amino acid will break, and then the transfer RNA will go off to get another amino acid, okay? Anyway, as that happens, it just happens over and over and over. Then here you can see the growing polypeptide chain getting larger and larger. Okay, and what happens then is that after termination, after the transfer RNAs encounter or the release factors encounter the stop codons, UAA, UAG, or UGA, then the whole complex comes apart. The small subunit of the ribosome, the large subunit, plus, and the polypeptide as they go, the transfer RNAs just go back to doing their regular business. Okay, so generally transcription is when you have RNA forming from DNA, because remember this is still in the language of nucleic acids, but when you have messenger RNA coding for the making of protein, it's called translation. It's sort of like you're taking the language of nucleic acids and translating it into the language of proteins. Okay, that's why one of the ways that you can kind of remember. After the whole complex comes apart, the polypeptide, if there's only one polypeptide, of a particular protein, it will start doing the bending and folding due to the R groups, right, of the amino acids, hydrogen bonding with different R groups, or being repelled from each other, so they'd be more toward the outside of the molecule. And of course, the shape of the molecule actually relates to its function. If the enzyme or any other kind of protein doesn't have the right configuration, oftentimes the enzyme or whatever other kind of protein it might not function as it should. Okay, that means that you have a non-functional protein. That means that it can't do its job. Okay? Okay? Anyway, so an active protein has the right conformation, all right? And the right binding sites. So let's go over that again. Okay, I wanted to tell you about that when we were looking at the diagram. But now we have transcription that happens in the cell nucleus. Okay? This doesn't happen in bacterial cells. It happens in only what we call eukaryotic cells, such as plants and animals, protists, and fungi. First of all, messenger RNA is transcribed from a DNA template, right? A double-stranded DNA unwinds, and it's kind of helped along by something called RNA polymerase, which is an enzyme, okay? Kind of helps to open it up, all right? So when you think about this, so mRNA transcribed from the gene on DNA. What would be the complementary base sequence on the following DNA template? So here you have GAA, CTG, ATT. Okay, think about that. Okay, then we'll do it right now. You'll have a C, here you have a U, right? Remember U is in RNA, right? Don't let that trip you up. Don't let me trick you, okay? Then we have U pairing with A, then we have G pairing with C, A pairing with T, C pairing with G, U pairing with A, and then we have two A's. Okay, so you have complementary mRNA sequences. Okay, so this is another thing. You might also have a test question on this, so please don't get confused. Remember, DNA has T, okay, RNA has U, okay? So the next thing happens is that the non-coding regions on the messenger RNA are spliced out, okay? Then the coding regions of the messenger RNA, called the exons, are spliced together. Then you have a functional, ready-to-go messenger RNA. Okay. Now translation, of course, so the messenger RNA leaves the nucleus and translation happens in the cytoplasm of the cell. First of all, the mRNA will join the small subunit of the ribosome. Okay. The tRNA will attach, and so will the large subunit of the ribosome. The tRNA anticodon, remember that's just a series of 3 complementary bases that pair with the codon on the messenger RNA. So if the codon on the messenger RNA was CAU, yeah, which codes for histidine, the anticodon, the complementary bases of the anticodon would of course be GUA, right? It's the same thing, okay? So this is is really fascinating. Another thing is that it seems very simple when you really think about it. It's very simple, but it's so remarkable, literally remarkable, something to remark upon, you know? So this tRNA will bring in, on the other side of the molecule, will bring in the amino acid, whatever amino acid that the codon will code for. Then you'll have a second transfer RNA bringing in amino acid number 2. Okay. Then between amino acid number 1 and amino acid number 2, you'll have the formation of a peptide bond. Okay. The first and second amino acids will bond together with each other. Okay. Alrighty. Then what happens next is the first tRNA will leave. It'll actually take on off. Alright, it'll actually go away. Alright? And then the ribosome itself, it's a funny thing. A lot of people don't know about typewriters nowadays, but I was a secretary for a while, you know, at least an office worker, right? So what usually, if you're typing on a typewriter, the really old typewriters would actually have a ribbon that would move, you know, as you're typing. Then what happens is that the typewriter and the ribbon moves as you're typing along. So you can kind of think of the messenger RNA almost like the ribbon, and you can think of the ribosome actually as being like the typewriter. But in this case, it's inverse. The messenger RNA stays there, and the ribosome zips along the messenger RNA, and it happens really fast. Okay, so the ribosome will then move over to the next, you know, to the next codon. Okay, and the next thing that would happen is that the third tRNA will bring in the third amino acid, peptide bond between the second and the third amino acid. Okay, and the second tRNA leaves, and this happens over and over and over from 50 to 500 times or more. Okay, and then this continues all those times until finally the codon that signals the cell to stop is actually encountered. Okay, then the whole complex comes apart, and then everybody goes on about their business. Okay, alrighty. So let's go over that again, because I know that it's not easy material to know. Okay, so don't get stressed out. Don't worry about it so much. You know, you just review. You got to go over it again. Okay, and again, and that'll help you to remember what's happening. Okay, first of all, you have a sequence or a section of the double-stranded DNA. Usually what happens is it unwinds a little bit, and then the bases, okay, and it's kind of helped along by an enzyme known as RNA polymerase. The bases for the RNA come in, and they'll actually use the DNA as a template, forming messenger RNA, okay, a sequence of nucleic or nucleotides making up the RNA molecule, just like nucleotides make up DNA. Okay, then you have the introns and non-coding regions of the messenger RNA spliced out. The exons, or the part of the messenger RNA that codes for a product, will be spliced together. Okay, so here you have an adult or a mature messenger RNA all ready to go. Okay, then it's sort of like getting ready to go out. Okay, anyway, so this messenger RNA then leaves its home, the home of the DNA, goes out into the cytoplasm. okay, then what happens in it forms a complex between the small subunit of the ribosome. So you have the mRNA forming a complex with a small subunit. The transfer RNA comes on in, okay, and then the large subunit of the ribosome comes in too, and they form this complex. The first transfer RNA will have the anticodon, which binds on to the codon, okay, of the messenger RNA. All right. The other side of the transfer RNA has an amino acid, right? The amino acid, okay, is there then, all right? Then what happens is that a second transfer RNA comes in, okay, binds with its anticodon onto the next triplet of codons on the messenger RNA. It also has brought in on the other side of the molecule another amino acid, all right? Then the two amino acids, a peptide bond forms between the two amino acids. Then the first transfer RNA goes off to get another amino acid, depending on what's going on in the cell, because this is happening all over inside of the cell all the time, okay? A third transfer RNA comes in with its anticodon, which matches with the codon, the next triplet of bases on the codon on the messenger RNA. It also brings in another amino acid, and then you can see here that this is actually showing the whole process. So as the ribosome is zipping along this messenger RNA, it happens very quickly. You have the transfer RNA coming in, the peptide bond being formed, the transfer RNA going out, the next transfer RNA coming in, and the thing is that the ribosome is actually moving, okay, along the messenger RNA. So it's pretty interesting, and it is like a miracle that all the living things, practically all living things, cells can do this, because even bacteria need to have proteins as well, even though they have a little bit different method of actually doing protein synthesis. Okay. Anyway, then of course, finally, after the ribosome encounter or the transfer RNA or the release factors comes upon a stop codon, then the whole complex comes apart, and then the polypeptide bends and folds in the first level of protein structure, which is just a sequence of amino acid making up the protein, usually between 50 and 500 amino acids for one polypeptide. Second level of protein structure is when you have nearby R groups of the different amino acids reacting to each other, kind of folding into a beta sheet or an alpha helix or a random coil. Third level of protein structure, when far apart, okay, amino acids or the R groups on the amino acid forms like the general three-dimensional shape of the polypeptide. Okay, and then if you have fourth level of protein structure, because not all proteins have all four levels, okay, then you'll have more than one polypeptide forming a particular protein. Hemoglobin is a good example of that. Hemoglobin actually has 4 polypeptides, and hemoglobin, what does it do? It actually helps to carry oxygen, okay, in our blood to where it's needed. All right. It also takes away carbon dioxide. All right, anyway, so then you have the three-dimensional shape of the protein, right? And generally, when the protein is activated, okay, then it can actually do its job, all right? Okay, so that kind of talks about that, okay? The next thing we're going to talk about is mutation. And mutation, of course, is the stuff of evolution. Okay, we're going to talk about evolution a few lectures from now, okay? So mutation, pretty much, all right, is any kind of change in DNA, okay? And one of the things to remember, any kind of aspect of a particular plant, okay? Any kind of aspect of an animal, any aspect that is actually physically present, okay, in any kind of organism is coded for in the DNA. The DNA in our bodies codes for every trait, such as even eye color. Okay, this looks like a lady's eyes, yeah? She has all these long eyelashes and like that. Okay, eye color is something that is probably controlled by more than one set of genes. Remember when we talked about genetics, we talked about polygenic inheritance, such as skin color? Well, eye color is probably the same thing. Another kind of aspect in our human bodies that is kind of governed by more than one set of genes, there are probably many sets, is height. Okay, it's another one. You have maybe at least 3 sets of genes that will code for how tall you are. Okay, so that can be interesting things. It's always interesting, genetics. Students really, really like that because then you can kind of see what, each other has, So in humans, we actually have like a single gene that will code for something like if your little finger is bent inward, Okay, that's, I think, is a dominant trait. Okay, another trait too is that whether your earlobe is attached to the side of your neck. Okay, so you have unattached earlobes, you have attached earlobes as well. You also have the presence or absence of a widow's peak. A widow's peak is just like when your hairline comes down like this. Like I have a widow's peak, I think it's a dominant allele, all right? Another kind of thing that people have too is freckles. Freckles is dominant to having no freckles, but people who don't have freckles are actually, there are more people in the population that don't have freckles than do, okay? So just because a trait is dominant doesn't mean that it's the most widespread of the types in the population, okay? So if we go back to the slides right now, the PowerPoint, we can also see this is unfortunate and it's not a happy thing, but you can also see that mutation that is a change in DNA can actually also result in something like this. This is like a normal cell replicating or undergoing mitosis, and then this cell here should be identical to this cell, but then what happens is that when this cell does its division, there might have been a mutation occurring in occurring either in its germline or in its daughter cell. So this looks like mitosis. It doesn't have the recombination aspect, but here, this is actually an abnormal cell, and I believe that this is actually depicting a cancer cell. Okay, so what is cancer? Cancer pretty much in humans, pretty much, and in any kind of animal, is when you have mitosis or cell division kind of gone crazy. Okay, what happens is that in cancer, you actually have cells, so you have like one cell that a mutation has happened in, becoming a cancer cell. What happens is that they divide, and they just keep dividing and keep dividing and keep dividing. Cancer cells do not behave like normal cells. Normal cells, if you put normal cells in a petri dish, okay, and you let them divide and divide and divide, as soon as the surface is covered, they'll stop dividing. Not just that, is that I believe that most cells that are normal will usually stop dividing after maybe 50 times, maybe. I might be having that wrong. But there is a time in the life of a normal cell where it doesn't divide anymore, okay? Cancer cells are not like that. Theoretically, they can keep dividing forever. Okay? So what they'll do is that what we call cancer is when you have abnormal cells crowding out normal cells in the body, and they start, when cancer undergoes metastasis, what happens is that it will actually break off from the original cancerous tumor and go to other areas of somebody's body. One thing I want to tell you is that, just because cancer is pretty, all of us know people who've either had cancer or maybe had died of cancer, but just because that is true doesn't mean that anybody has to get cancer. Even though we're living longer and there are mutagens or substances in our environment that can cause mutations, it doesn't mean that people have to get cancer. And even if you do get cancer, it doesn't mean you have to get really sick or that you have to die from it. There's lots of good advanced ways to treat cancers, okay? So cancer is just a state of the body. It's not anything necessarily to be afraid of, even though, of course, if you see somebody really sick with cancer, it can be very, can scare you, okay? And it's not a pleasant thing to experience, for sure. But more and more, if you can catch abnormalities early, okay, if you can catch it early, they can usually excise it with surgery and treat you for it, and you'll survive. I have a friend who was diagnosed with breast cancer last year, and she's okay now. Okay, she had a little bit of surgery, but it was not that invasive as far as I could tell. She had radiation therapy, and she's cancer-free, as far as we know now, because they have blood tests that can tell you, whether you're still cancerous or not. So just because, just because there is a thing such as cancer, doesn't mean that you have to get it. Okay, if you do happen to be unfortunate enough to get it, it doesn't mean that you have to get really, really sick or die from it. Okay, just remember that. So it's not really something to be afraid of. Okay, so mutation. Okay, so mutations can be of different types. First of all, you have something called point mutation. Okay, that usually is a more minor of the different kinds of mutations. Okay, because all that happens is, like we saw a little bit earlier, like when you have a T substituting for a C, that means you have a cytosine pairing with an adenine. Okay, or sometimes you can have an enol form, and then you can actually have a C substituting for a T, or you have a cytosine pairing with an adenine. I'm getting confused here, I think. Anyway, so if you were to look at the genetic code, in your textbook, you can see that maybe UAU and UAC both code for tyrosine. Okay, so you can actually have a mutation to UAU, and if it mutates to UAG or UAC, that is actually what they call a silent mutation, because it doesn't have any kind of effect in what kind of protein or what kind of amino acid is actually coded for. Okay, so base substitution tends to be the smallest kind of mutation or change in DNA, and it is actually one that tends to be sometimes is not harmful. Okay, even though there is one case where a base substitution actually can bring about very detrimental effects on the organism, and that of course is what happens when you have a base substitution that codes for valine instead. And people who have this base substitution can have sickle cell trait, or they'll carry a cell or genes for sickle cell trait, and that can be very serious, because if a child has 2 copies of the sickle cell trait, they'll have sickle cell disease that in the past has oftentimes ended up with a person passing away fairly young. Okay, because what that calls for is it accounts for abnormal sickling in the hemoglobin in conditions of low oxygen, such as a person who might have sickle cell trait. If they go up to high elevation, sometimes you can have the sickling happen, and it actually causes a whole array of symptoms, and also can make the person really, really sick. It's really very bad. Okay, so that's one instance where a single mutation, a single base substitution can cause disease. Right? Another kind of mutation is called a frame shift mutation. Remember that generally the codons are read in groups of 3s, triplets of 3s. If you have a frame shift mutation, what that can happen is you can have something called a deletion. Okay, so if you were to actually depict a deletion in terms of something that makes sense to us, such as these triplets, the rat saw the cat. Okay, if the H, okay, was excised out, everything downstream from that mutation where you had a deletion, it's like, mumbo jumbo. It doesn't make any kind of sense. And that can happen when you have a frame shift mutation. So frame shift mutations can also be very serious. Okay, so remember, so you always have this triplet bases read in triplets. So if you have a deletion or if you have an insertion, you can just have mumbo jumbo further down. So the rat saw the cat, you have an insertion. So you have all this stuff that doesn't make any kind of sense. So you can actually have the coding for something that doesn't code for anything that you might actually want. Okay, very bad. So these deletions and insertions can be as serious as the one instance of the point mutation, which is very serious. It can cause even, the death of the organism. Okay. Now, another way to classify mutations, sometimes you have something called a somatic mutation. So somatic mutation happens in the body cells of the organisms, and in plants, the somatic mutation can occur in the root or the stem or in the leaves. Okay? And if we go to the Elmo right now, we can see a kind of mutation that has happened in this plant. This is a leaf of a ficus. And you can see it's very attractive because we humans, we like this because it's nice looking and it's unusual. What do you see? Right? You see a leaf that has 2 different shades of green plus an area that doesn't have any coloration of it at all. Okay? So in this leaf, you have something called variegation. Okay, variegation. That means you have more than one color of the leaf. For us humans, that's a really nice point or feature of an ornamental plant. But, okay, if you actually look at it, okay, this area doesn't have any chlorophyll in it. So when this happens in nature, usually it's not good for the plant, because if you have less chlorophyll, you have less photosynthesis. And oftentimes a plant that has all this variegation, you can actually have the plant being stunted, okay? Now another thing too, in another plant, you can actually see this happens in different plants. Okay, so you can see it's very attractive horticulturally, or when we want to grow ornamental plants. But generally in these plants too, it's not that good of a thing necessarily in nature. Okay, so you can have a somatic mutation. Another thing about the somatic mutation is that any kind of offspring that are produced asexually can be affected that way. Everybody's probably seen when you have variegated hibiscus where the leaves are kind of whitish and yellowish and green, and then you can actually see a sport where you have a little bit of the plant reverting back to its full green color. When you have an organism that has 2 different types of body cells, that's called a chimera. Okay, a chimera in Greek mythology was like an animal that was a composite animal made up of different parts of different kinds of animals. Okay? Now sometimes when you have a mutation that's happening during gamete formation, you can also oftentimes have this mutation being passed on to its offspring. And maybe you can have new allele, okay, in a population. And the thing that's important about this is that mutation is pretty much the raw material for what we call evolution, or changes in organisms. So that's what you call a speciation, when you have new species rising from old species. This is exciting. Most mutations are not good. Okay, it's sort of like taking an engine that's working pretty well and going in and loosening one screw or one bolt and kind of expecting the machine to work better. That doesn't happen. Usually it's deleterious or not good for the offspring, but it is a raw stuff of evolution. Okay, now we're going to come to the summary of our talk today. All right, for one thing, we went over deoxyribonucleic acid, the stuff of which our bodies are made. Our DNA codes for all parts of our bodies, all the chemicals that we produce, all the different personality traits are probably encoded for in our DNA. Okay, we talked about Watson and Crick. Okay, also Rosalind Franklin. We also talked a little about Morris Wilkins, and we talked about Beadle and Tatum, and also Hershey and Chase. Okay, all these brilliant minds that led to the knowledge of the structure of the double helix. We talked about the genetic code, okay, written in triplets of bases. We talked about protein synthesis, transcription, and translation. And then lastly, we finished off with mutations, the stuff of evolution. Okay? Okay, that's all we have time for today. We're quickly running out of time, and hope that you enjoyed today's lecture. Don't let it overwhelm you, okay? And study hard, and always email me if you have questions, and we'll see you next time. All right? Bye-bye.